What is the relationship between telomere lengths and cancer, heart disease, high blood pressure, infertility, and even rheumatoid arthritis? You will be surprised with the findings as I will be sharing various studies with you today. Hi, my name is William and welcome to my channel, where I share science-based evidences on products claimed and effective methods related to health and beauty concerns, so you can make an informed buying power. If you're new to my channel, I upload a new video every Friday, so please subscribe if you haven't and click on the notification bell to get notified of my new videos. In my previous videos, I talked about using telomere lengths and gene expression to measure our body's actual biological age. If you have missed out, there's a link below in the description box or click on the card above to watch it. All references and cited research articles will be also provided in the description below for any one of you who are interested to read further. In today's topic, I'll be sharing how telomere shortening affects various lifestyle diseases. We start with breast cancer. Researchers have found that telomere shortening is a common event involved in malignant transformation. Critically short telomeres may trigger chromosomal anomaly and produce genomic instability leading to cancer development. To understand the role of telomere length in breast cancer causes, this study measured the length of individual telomere thickness in single cells from hereditary and sporadic breast cancers. Hereditary cancer is caused by gene mutations passed on from parents to their children, whereas sporadic cancer arises from gene damage acquired from environmental and various factors. A total of 104 tumor tissue samples from 75 hereditary breast tumors and 29 sporadic tumors were analyzed. It was found that hereditary tumors were characterized by shorter telomere length compared to sporadic tumors. Short telomeres also correlated with a type of tumor called estrogen receptor negative tumors in sporadic cases, but not in hereditary cases. In addition, telomere lengths help to define a subset of hereditary tumors with short telomeres associated with increased expression of anti-apoptotic proteins, which means the cancer lived longer than usual. These findings highlight the potential interest of telomere measurements as markers of aggressiveness in breast cancer. In general, this study concluded that short telomeres are frequent in hereditary breast tumors and are associated with high tumor grade. Let's look at heart disease. This review paper states that in the majority of studies, patients with early onset coronary heart diseases have been shown to have shorter telomere lengths in a type of white blood cells called leukocytes. In addition, patients with diabetes mellitus complications tend to have shorter leukocyte telomere lengths than control subjects. Another paper studied the relationship between telomere lengths and heart disease in type 2 diabetes mellitus patients. It was found that vascular aging signs were more prominent in patients with diabetes. However, despite diabetes, vascular changes in patients with long telomeres were very modest and were very similar to the vascular walls in healthy individuals, thus suggesting long lymphocyte telomeres may have a protective effect on the vascular wall and may prevent vascular wall deterioration caused by glucose metabolism disorders. As for the case of hypertension, this study was to investigate the association between leukocyte telomere lengths and the risk and prognosis of hypertension in a Chinese population. The relative telomere length of leukocytes were determined in 767 subjects, 379 healthy controls, and 388 hypertensive patients aged between 30 and 80 years old. The median telomere length ratio was shorter in hypertensive than in healthy normotensive subjects. After 5 years of follow-up, subjects with shorter telomeres were at higher risk of developing coronary artery disease than individuals with longer telomere lengths. The data suggests that mean leukocyte telomere length is a potential predictor of coronary artery disease and support the hypothesis that differences in biological aging can contribute to the risk and variability of developing hypertension and cardiovascular diseases. Whereas another study in an Indian population concluded a similar finding as well. Next, we discuss about infertility. In this pilot study, 32 infertile men with unknown causes and 25 controls were analyzed for sperm telomere length and correlated it with sperm DNA fragmentation index and reactive oxygen species levels. The relative sperm mean telomere length of infertile men was found to be significantly lower compared to controls. It is concluded that shorter telomeres in sperm may be one of the causative factors responsible for male infertility and suggests further detailed studies to confirm these findings. Whereas for women, this current research is aimed at finding potential non-invasive biomarkers that will help us learn more about the mechanisms at play in failed assisted reproduction treatment. This pilot study examined the relationship between cell-free DNA in plasma and telomere length in lymphocytes among women undergoing in vitro fertilization, or popularly known as IVF, and compared this to a healthy control group. 20 IVF women and 10 control women were taken as subjects. Women undergoing IVF had statistically significant higher levels of cell-free DNA and shorter telomeres compared to healthy controls. So, can it be suggestive that infertile women have shorter telomere lengths? Something for us to ponder about. Finally, for rheumatoid arthritis relationship, white blood cell and telomere length were measured from 176 hospital-based rheumatoid arthritis cases and from 1,151 controls. Telomere length were compared between cases and controls, and the effects of disease duration, severity, and HLA-DRB1 alleles, which is the gene associated with an increased incidence of rheumatoid arthritis, were assessed. A reduced telomere length in a large group of patients with rheumatoid arthritis compared with controls has been found. The reduction is apparently independent of disease duration and markers of disease severity, but is influenced by HLA-DRB1 genotype. So, can we conclude that short telomere length causes most age-related diseases? Yes, many articles seem to say so, but is it all diseases? No, because the science of aging is complicated and further studies are still needed to understand them clearly, especially on the human race. But it is enough to conclude that to age more gracefully, we should prevent telomeres from shortening too fast. So what are the ways? Find out next week.